peel back the layers on something that honestly really captured my interest when I first read about it. The dark fermentation metabolic pathway. It's this natural process, and it holds just incredible potential for producing something called biohydrogen, a clean energy source. We've got some really detailed source material today, a deep exploration from a recent research paper that kind of lays out this whole intricate biological dance.
Yeah, it's fascinating stuff. And our mission, really, in this deep dive is to break it down. Explain clearly and simply how this process works, from the start, the inputs, all the way to the critical outputs. We'll try to highlight the nuanced steps and the conditions that really shape how efficient it is. And we'll break down why certain metabolic routes give you different results for biohydrogen. So you understand not just what's happening inside the cell, but, well, why it matters for our energy future.
Right. So let's explore this core process. How microorganisms can basically be harnessed to create a sustainable energy source. It's quite a journey, deep to cellular chemistry. So to really get a grip on this, we need to start with the basics. What exactly is dark fermentation? At its core, it's an anaerobic process, meaning it happens without oxygen.
Exactly. No oxygen and no light either, hence the dark part. And its core purpose. It's all about breaking down organic compounds. These microbes, they take in organic food, essentially, and when they break it down without oxygen, they end up with, well, extra electrons. They need to dump those electrons somewhere. So they grab protons, just loose hydrogen ions floating around, add the electrons, and boom, hydrogen gas. bioH₂.
And hydrogen isn't the only thing they make, right?
Not at all. You also get other byproducts, things like alcohols, CO2, and various organic acids. It's quite a mix.
Okay, so these microbes aren't too picky. They can eat various things like carbohydrates, sugars, proteins, fats.
Pretty much, yeah. They're versatile. But to make it easier to follow the actual biochemistry, the pathway is usually shown using glucose as the model. It's just simpler to track.
Makes sense. So let's start there with glucose. What's the absolute first step?
Okay, first step. Glucose gets converted into pyruvate. This happens through a really fundamental process called glycolysis. You probably remember that from biology class.
Vaguely. Glycolysis, right. And this initial breakdown, it doesn't just create pyruvate. It also generates a couple of other key things for later. NADH, which is like a temporary electron carrier.
An electron taxi, you called it before.
Yeah, exactly.
Sure.
An electron taxi, and also ATP, which is the cell's direct energy money.
Got it. So pyruvate is formed. What happens next? You mentioned it's a key junction.
It really is.
All right.
Pyruvate sits at this metabolic crossroads. From here, depending on the specific bug we're talking about, the pyruvate gets converted into something called acetyl-CoA. And this conversion uses one of two main enzymes. And which enzyme it uses really dictates the whole rest of the story for hydrogen production. Okay, so two diverging paths right from this pyruvate point. What are these two key enzymes?
Right. First, there's Pyruvate Formate Lyase, or PFL. You tend to find this more in facultative anaerobes. Those are the microbes that can kind of swish hit, live with or without oxygen, though you do find it in some strict anaerobes too, like certain Clostridium species.
And what's notable about this PFL route?
The key thing here is it doesn't immediately generate those electron carriers, those taxis, that are needed directly for making hydrogen gas right away.
Okay, so that's PFL. What's the other enzyme?
The other one is Pyruvate Ferredoxin Oxidoreductase, PFOR for short. This one is the go-to for strict anaerobes, the ones that absolutely cannot tolerate oxygen. And this PFOR enzyme is directly involved in activating the molecules that make hydrogen.
And those are the hydrogenases you mentioned earlier.
Exactly. Hydrogenases. They're the actual catalysts, the little molecular machines that churn out the bioH₂. And there are two main types relevant here. FeFe hydrogenases, you find those linked to both the PFL and PFOR pathways. And then there are NiFe hydrogenases. These are a bit more robust, more tolerant to oxygen, so you typically only see them associated with that PFL pathway in the facultative microbes.
Okay, let's follow that first path then. The PFL pathway, the one common in facultative anaerobes. Pyruvate gets hit by PFL.
And it splits into two things, acetyl-CoA, which we'll come back to, and formate.
Formate, okay. What happens to the formate?
The formate gets oxidized by another enzyme complex called formate hydrogen lyase, FHL. This step activates the hydrogenase enzyme, and then that enzyme does the final job. It takes protons, H+, and reduces them to H2 gas. You also get CO2 released here.
So how much hydrogen can you theoretically get from this pathway?
Well, since the hydrogen comes from the formate, and you get maximum 2 formates from 1 glucose, the theoretical maximum yield here is 2 moles of bioH₂ per mole of glucose.
2 moles per mole. Okay. Seems straightforward.
Pretty much, yeah.
And the acetyl-CoA that was also produced. What's its fate in this PFL pathway?
Right. The acetyl-CoA, it gets processed further, typically oxidized to acetate. And this process is actually useful for the cell because it generates more ATP, more energy.
Okay, but you mentioned something crucial earlier about NADH in this pathway.
Yes, absolutely critical. Remember that NADH taxi made during glycolysis?
Yeah.
In this PFL pathway, that NADH cannot be used to make more hydrogen. It's just not wired that way metabolically. So the cell has to regenerate its NAD plus supply somehow. It needs to offload those electrons from NADH onto something else.
And what does it use?
Typically, it uses them to produce a reduced organic compound. Ethanol is a very common one. It takes a couple more enzyme steps, basically converting acetyl-CoA eventually into ethanol.
So it's a trade-off. They regenerate NAD plus to keep glycolysis going, but instead of extra hydrogen, they make ethanol.
Precisely. You get ethanol, which can be valuable, but you're capped at those 2 moles of H2 from the formate.
Okay, now let's switch gears. What about the other route? The PFOR pathway, used by the strict anaerobes. Here, you said bioH₂ production happens when pyruvate is converted to acetyl-CoA, but it involves ferredoxin.
Exactly. PFOR converts pyruvate to acetyl-CoA, but instead of formate, it produces CO2 and reduced ferredoxin. And reduced ferredoxin is a very strong electron donor. It can directly pass electrons to that FeFe hydrogenase, which then, just like before, reduces protons to make H2. So initially, this pathway also gives you 2 moles of bioH₂ per mole of glucose.
Okay, so far same yield, 2 moles. But you hinted this pathway has a trick up its sleeve. This is where it gets really interesting.
Yes. This is the crucial difference. Unlike the PFL pathway, the PFOR pathway, under the right conditions, can do something amazing with that NADH from glycolysis.
So it can use the NADH for hydrogen?
Potentially, yes, under ideal conditions, specifically low partial pressure of hydrogen.
Meaning when there isn't much hydrogen gas already built up in the system.
Exactly. When the conditions are favorable, the acetyl-CoA can still go to acetate, making ATP. But crucially, those 2 NADH molecules from glycolysis can also be used. They get reoxidized by donating their electrons via ferredoxin, usually, to other hydrogenases, specific NADH-dependent ones. And this process generates 2 more molecules of H2.
Wow, okay, so the initial 2 moles from pyruvate to acetyl-CoA conversion, plus another 2 moles from oxidizing the NADH.
That's it, giving you a theoretical maximum of up to 4 moles of bioH₂ per mole of glucose, double the yield of the PFL pathway.
Double. That's a huge difference. So what determines if you get 2 moles or 4 moles in this PFOR pathway? You mentioned conditions.
It really hinges on that hydrogen partial pressure. Making hydrogen from NADH is only thermodynamically favorable, meaning it only really wants to happen when the concentration of H2 already present is low.
Like an equilibrium thing.
Sort of, yeah. Think of it like trying to push more people into an already crowded room. It gets harder. As fermentation proceeds, the bioH₂ builds up. Partial pressure increases. Once that pressure gets too high, oxidizing NADH to make more H2 just isn't favorable anymore. The cell stops doing it efficiently.
So under those higher pressure conditions, what happens instead? The cell still needs to deal with the acetyl-CoA and the NADH.
It does, and so it shifts gears. Instead of maximizing hydrogen, it starts using the acetyl-CoA and the electrons from NADH to produce other, more reduced organic compounds.
So different byproducts again.
Right. In strict anaerobic fermentation, under high H2 pressure, you typically see things like butyric acid, maybe butanol, acetone. These become the main destinations for those electrons.
Which, as you mentioned, might actually have their own value, but it means less hydrogen fuel.
Exactly. It's a metabolic switch based on the environment inside the reactor.
Okay, so managing that hydrogen level seems key. But stepping back, regardless of PFL or PFOR, are there common challenges faced in any dark fermentation process?
Oh, definitely. One universal issue is that need to constantly reoxidize NADH. Whichever pathway is used, the cell must regenerate NAD+ to keep glycolysis running. If that stops, everything grinds to a halt. Maintaining that redox balance is fundamental.
Makes sense. And another issue people often talk about is pH drop, right? Acidification.
Yes, that's a major one. Because these fermentation processes often don't completely break down the starting organic matter, you get an accumulation of volatile fatty acids, VFAs.
Like the acetate and butyric acid we mentioned.
Exactly. And as these acids build up, they lower the pH of the culture medium.
And that's bad for the microbes.
Yeah, it can be really inhibitory. Most of these hydrogen-producing microorganisms don't like highly acidic conditions. It messes with their enzymes, their membranes, it slows down or even stops the hydrogen production. It's a significant practical challenge.
So how do researchers deal with it? Just keep adding base to neutralize the acid?
That's one approach, pH control. But there are other strategies too. One clever idea is to use the effluent, the liquid output from the dark fermentation reactor, which is full of these VFAs, and use it as the input for a second biological stage. For example, feeding it to photosynthetic bacteria for photofermentation, which can also produce hydrogen, or using it in microbial electrolysis cells, sort of a two-step process.
Turning a problem into a resource for the next step?
Exactly. Or another angle is to actually recover and purify those VFAs from the effluent. Things like acetic acid and butyric acid have commercial value themselves.
So you could potentially sell the acids as well as making hydrogen?
Potentially, yes. It adds another value stream to the whole process, makes the economics look a bit better.
These microbes are incredibly versatile. We focused on the PFL and PFOR pathways from glucose, but are there other completely different metabolic strategies microbes use to make biohydrogen?
Oh, absolutely. Nature's found many ways. For instance, there's the cellulolytic pathway. This involves microbes that can actually tackle really tough starting materials, like lignocellulosic phytomass, think wood chips or agricultural waste.
Stuff that's hard to break down.
Very hard. These microbes first have enzymes to break down the cellulose into simpler sugars, like glucose, and then they ferment those sugars to H2, plus the usual suspects like acetic and butyric acids and CO2.
What's the catch with that one? Sounds great using waste materials.
The main challenge is that breaking down the cellulose itself, the hydrolysis step, is often very slow. And the lignin component in plant matter makes it even more resistant. So you often need significant, sometimes costly pre-treatment of the biomass before the microbes can even get started.
Right, makes sense. Any other major pathways?
Well, we can also talk about pathways named for their main acid byproduct. There's the butyric pathway. Here, glucose gets converted, yielding roughly 2 moles of H2, 1 mole of butyric acid, CO2, and protons.
Limitation mean.
Again, the buildup of the acid, butyric acid in this case, causes pH drop, inhibits the microbes. Though, interestingly, the internal electron balance in this pathway can sometimes be slightly more favorable for H2 production compared to, say, the purely acetic pathway.
And speaking of which, there's an acetic pathway too.
Yep. Carried out by microbes like Clostridium aceticum. Theoretically, this pathway can convert glucose into 4 moles of H2, similar to the high-yield PFOR pathway, along with acetate, CO2, and protons.
So another route to potentially high yields.
Theoretically, yes. But again, the limitation is often the accumulation of acetic acid, which affects the microbes. And even though the theory says 4 moles of H2, in practice, it can be less efficient. We still produce CO2, and maintaining that crucial redox balance while forming acetate consumes some of the electrons that could have gone to hydrogen. So it's often a trade-off in reality.
It really highlights how many different biochemical solutions microbes have evolved, each with its own advantages and drawbacks.
It truly does.
Okay, wow. We've certainly journeyed deep into the world of dark fermentation today. It's really clear that, while the basic idea is simple, turn organic stuff into biohydrogen, the actual process, the efficiency, well, it all comes down to these molecular details and the environmental conditions, doesn't it?
Absolutely. What I find really fascinating is how these subtle shifts, like which enzyme gets used, PFL or PFOR, or just how much hydrogen gas is currently in the reactor, can completely change the outcome. It impacts not just how much hydrogen you get, but also what kinds of other byproducts made, which might be valuable too. And if you zoom out, it just underscores the incredible complexity and adaptability of microbial metabolism. It gives us so many different angles to potentially explore for sustainable energy.
It really does. And that complexity leads me to a final thought, maybe a question for you, our listener, to chew on. Given this intricate balancing act, the different pathways, the useful but sometimes problematic byproducts, the critical environmental factors like pH and hydrogen pressure, what do you think is the single most critical hurdle we need to overcome to make dark fermentation a truly widespread, efficient, and maybe economically viable source of renewable energy? Something to think about.